Hello, my dear students. This is again Dr. Siddalinga Murthy SM, Head of the Department of Botany, ADB First Grade College, Harpalli. And this video class is for B.Sc second semester. And this is of course the last video class. As I told you in the previous class, today I am going to tell you about Rhynia and Calamittis. Okay. So let us see the characters of Rhynia and the Calamittis. Yes, well students, as I told you in today's video class, let us discuss about the two important fossils which are included in your syllabus. So Rhynia, you can observe the plant. This Rhynia, it is a fossil plant. You can guess this fossil plant resemble like one of a living pteridophytic plant which you have already learnt. So by looking this one, a leafless pteridophyte, what you learnt already. So that is nothing but what? Xylotum. So this plant is similar to that of Xylotum. So this Rhynia plant in fact obtained in Scotland in one of the place called Rhynie. So therefore this plant is named as Rhynia. So there are two species of Rhynia, Rhynia major and Rhynia guani-vagani. So small differences there between the Rhynia major and Rhynia guani-vagani. Okay. So the plant obtained here is a sporophyte. You already know the meaning of sporophyte. So the plant body is differentiated into root, stem and leaf. So like that of the Xylotum, here you will find the rhizoids. And this is the horizontally growing rhizome, and these are the aerial branches. So you can observe here there are no leaves, but adventitious branches are seen, and there are terminal sporangia. So in Xylotum you don't find a terminal, instead axillary synangium are there, but here terminal sporangia are present. A small difference is there between the Rhynia major and Rhynia guani-vagani. So the major variety is something taller than guani-vagani. So Rhynia major is about 50 centimeter in height and about 1.5 to 6 millimeter in diameter, while this Rhynia guani-vagani is about 20 centimeter in height and 1 to 3 millimeter in diameter. So as I said already, in both the species of Rhynia, the roots are absent, but the rhizoids are present. Okay, so that is the simple external morphology of Rhynia plant. So as usual, at the terminal parts of the branches, we will find the sporangia. So pear-shaped terminal sporangia are present at the tips. For the examination point of view, this TS of Rhynia stem is very important. So you can observe the diagram here. The transverse section of the Rhynia, so the fossil that is obtained here, it is possible to take the transverse section with the help of some equipment. So this is the TS, so possible to take the transverse section of rocks also. So this fossil rock transverse section has been taken, and you can observe here. So as usual, the transverse section of the Rhynia stem shows three regions. This is called epidermis, and here is the cortex and the central Stila region. Epidermis is simple, made up of single layer parenchymatous compactly arranged cells. Here and there certain openings are present, and these openings are nothing but stomata, which help in the exchange of gases. So you can understand that as the plant had no leaves, here the chlorenchymatous tissue, chlorophyll containing tissue are present in the cortex. So the major part of the photosynthetic region in the Rhynia plant probably this green stem. Next is cortex. Cortex is made up of parenchymatous cells. However, few cells are compactly arranged just below the epidermis, and they are somewhat bigger in size. And next to that layer, almost all the cells of the cortex are small, spherical, loosely arranged, very clear intercellular spaces. Probably they contain the chloroplast also, but in the fossil view, so not clear. The presence of air space, or the intercellular space and the stomata clearly indicates that this is the photosynthetic tissue. Of course, we can make out this cortex into two regions, the outer cortex and the inner cortex. Coming to the Stila region, otherwise called the central cylinder, the central cylinder is occupied by a protostele. Students, you already know the meaning of a protostele. A stele without pith is called protostele. A single xylem element is present, that is surrounded by few elements of phloem. So that type of simple protostele is present in the center. However, the structures like endodermis and pericycle not clear. So this is about the simple anatomy of Rhynia stem. As far as the reproduction is considered, the plant body is a sporophyte, so it reproduces asexually by forming the spores. The spores are produced inside the sporangium. As I said already, the sporangia are present at the terminal region, and inside this sporangia the spores are produced, and these spores after their liberation germinate and forms the new Rhynia plant. Here the plant is homosporous, that means only one type of spores are present. So as usual, the sporangium is pear-shaped structure. It contains the wall, otherwise called jacket. The inner wall is called tapetum, which supplies the food material to the developing spores. So the tissue present inside the sporangium undergo meiotic division. Haploid spores are produced, and these haploid spores after the liberation from the sporangia germinate and a gametophyte is formed. But the evidences for gametophyte in the form of fossils are not available. Okay, so that's all about the Rhynia. Students, if the question is asked for the 10 marks, you have to write both external morphology and TS of stem, as I said now, this one and this one. If the Rhynia stem is asked for 5 marks, then you have to draw only this diagram and explain only epidermis, cortex and central cylinder. So this is very important, TS of Rhynia stem. Okay. So now let us see about the second one, that is called as calamitis. So by looking at the diagram, you can understand the plant almost similar to that of one of the living plant, that is what? Equisetum. So even Equisetum also belongs to the class what? Spinopsida. So class Spinopsida, order Calamatilis and family Calamitiaceae. So as far as the external morphology of this plant is concerned, the plant as usual consists of rhizome, aerial axis and several branches. The rhizome is very stout. Nodes and internodes are present. From this underground horizontally growing rhizome, erect branches are produced. And these erect branches also contain nodes and internodes. The stem gives a kind of jointed appearance. So several branches are seen from that aerial branches. Roots are present particularly from the nodal region of the rhizome. And even from the aerial axis also, from the lower nodes, the roots are produced. And these roots provide additional support to the plant. As far as the transverse section of the stem is considered, it is more or less similar to that of the Equisetum. So we can observe here, this is the section of stem taken at the internodal region, and that is nothing but the Calamitis fossil. As usual, the transverse section shows epidermis, cortex and the Stila region. But here what happened? During the fossilization, only the hard part of the tissues got fossilized. The soft tissues are not stored here. So therefore, this entire region, what you observe is the cortical region. Here will be epidermis and there will be cortex. So cortex probably made up of parenchymatous tissue. So that is why it is not preserved. Only an empty area is present that indicates the cortex. Of course, leaves were present on the plant. So therefore, cortex made up of parenchymatous tissue. Coming to the Stila region, There will be endodermis and pericycle, but not clear in the fossil section. The main characteristic feature of the stem is the Stila region. Exactly the stele appears to that of equisetum. Several bundles of xylem and phloem are present in the form of a circular ring. So each vascular bundle is nothing but conjoint collateral vascular bundle. That is, phloem and xylem are present in the same bundle. Phloem is present towards the outer side, which is not fossilized because soft tissue, and the xylem is present towards the central region. So this xylem is preserved here. So the xylem here, it is very hard, indicating that this xylem is probably the secondary xylem. So the secondary xylem is made up of radial rows of tracheids. So the major part of the xylem is what? Tracheids. If you remember, the protoxylem elements of the vascular bundle break down and form a cavity. And that cavity or a canal, you called it as what? Carinal canal, this one. So the carinal canal found in the equisetum. So that type of carinal canals are present in the vascular bundles of the calamitous stem. This one, carinal canal. So the presence of the carinal canal clearly indicates that this is similar to that of what? Equisetum plant. Students, here a broad pith is present. So because of the presence of pith here, we can say that it is a siphonostele. In Rhinia we find protostele, and here we find a siphonostele. So pith is in the form of hollow cavity because it is hollow here, no parenchymatous tissue. However, if the section passed through the nodal region or the diaphragm, we can find a solid and stout pith. So secondary growth is not seen in the pteridophytes. Of course, in the living pteridophytes, the secondary growth or the secondary xylem is not present. But you observe here, in the fossil pteridophytes, we find the secondary xylem. That means what happens here? Earlier, there were some seed pteridophytes, otherwise woody pteridophytes, and even there were secondary growth also in the earlier days. But now, what the living pteridophytes we have, we don't see the secondary growth. So this is how xylem helps in the conduction of water and food material, as you know, water and minerals, and the phloem helps in the conduction of food materials. So that's all about the internal structure of calamitous stem. Students, you can expect the calamitous stem for the 5 marks. You can draw this diagram. This is a clear information, cellular view, but if you draw this diagram, it is enough. You have to draw this diagram and you can briefly explain epidermis, cortex and the Stila region, what I said so far. Okay, so that's all about the Rhinia and calamitous. Yes, that's all about Rhinia and calamitous. Students, hope you all have got the notes. So we have circulated the notes through your WhatsApp group. So hope all of you have received the notes. Students, if anybody did not receive the note, you can contact me. You have the WhatsApp number. So you call me and I will make you get the notes. So that's all about the things, whatever I want to tell you. So read well, prepare well. As soon as the lockdown is over, there will be examinations. So in the free time now, what you are having plenty of time. So in this time, you work hard, prepare well. So from the beginning to end, whatever the topics we have covered so far, and you go through the notes also, prepare well, and get ready for the coming examinations. So stay home, be safe, and I wish you all the best. Okay, thank you.